Okay, welcome back. This is the chromosomal basis of genetics, part 3, and in this section we're going to finish off chapter 15, or what we're going to cover in chapter 15. And so I'm going to move to our PowerPoint and we'll get started. I'm back in our, oops, let me fix this up here. I'm back in the storage room, and here we are. So we've covered some sex-linked traits in humans, and now we're going to take a look at how alterations in chromosome number and structure can cause some genetic disorders. This chapter, remember, is the chromosomal basis of genetics. So let's take a look at what happens if there's an alteration in chromosome number. So first of all, the term non-disjunction is going to be something we need to know. Non-disjunction is a situation in which chromosomes fail to separate properly during meiosis or during mitosis. There's kind of a mistake during mitosis or during meiosis in which chromosomes fail to separate properly. That results in a change in chromosome number. Here, let me get this going. Let me get my pen going here. Chromosome number, write the word number in there. Okay, so when there's a mistake or a failure to separate of the chromosomes, what that can lead to is a change in chromosome number. Sometimes that change is a small change. We call this an aneuploidy. That's on the next slide. That's a change that in chromosome number that affects maybe one or several chromosomes in a cell. A polyploidy is when you've got a change in chromosome number that affects many chromosomes, right? That's what the word poly means, whole sets of chromosomes. So we'll take a look at both of these. This is a picture of non-disjunction happening in two cells going through meiosis to make gametes. So these are our gametes down here, okay? We gotta make let's make them into sperm. So non-disjunction can result in an aneuploidy, a change in chromosome number that's a small change in chromosome number. And here's our picture of non-disjunction. Here we've got a cell going through, on the left over here, we've got a cell going through meiosis 1, and homologous chromosomes are supposed to separate. But oops, bright yellow star, they're not separating. There's non-disjunction happening, or failure to separate in the homologous chromosomes. And then the cell goes through meiosis 2, and this cell over here has an extra set of chromosomes separating, an extra chromosome, and this cell is missing a chromosome. And so the sperm that are made have either an extra chromosome, n plus 1 would be a haploid number plus 1, or a missing chromosome number. In humans, this would be something like our cells have 46 chromosomes, two sets of 23, and our gametes have 23 chromosomes. Okay? If a non-disjunction like this happened, we could have sperm that have maybe 22 chromosomes or 24 chromosomes in them. Over on the right, we've got non-disjunction happening in a different way. At meiosis 2, there's a problem. So in meiosis 1, the cell's homologous chromosomes separate perfectly. Oops, but in meiosis 2, one of these chromosomes fails to separate its sister chromatids properly. And now we've got some sperm that have an unusual chromosome number and some sperm that turn out okay. So and if these sperm meet up with an egg, fertilize that egg, then you've got an organism that has an extra chromosome in it, an aneuploidy. Are there any aneuploidies in humans? Yes. So we can specify the type of aneuploidy using these terms, trisomy and monosomy. Trisomy is 3, so having an extra chromosome. Monosomy, mono means 1, so missing a chromosome. And it turns out there aren't very many human disorders due to aneuploidies in autosomes. This is pretty rare, actually. But you do know one, and you know one well, and that is Down syndrome. People with Down syndrome have what's called trisomy 21. So if we look at the karyotype of this little girl, she has 3 copies of chromosome 21. Sometimes we call March 21 National Down Syndrome Day because 321. individuals with Down syndrome have 3 copies of chromosome 21. Most of you know someone with Down syndrome, and you probably get along with this person really well. People with Down syndrome have some delays. They, however, do very well in society and have productive quality life. And unfortunately, some people with Down syndrome have a higher risk of things like heart disease, hypothyroidism, leukemia, and then after age 50, an increased risk of dementia or Alzheimer's. In any case, there are 2 other trisomies in humans that result in live births occasionally. Now, most of the time, if you inherit an extra chromosome, an extra autosome, the fetus won't develop properly and is miscarried. Many, many, many of our miscarriages in humans are the result of an aneuploidy. But there are 2 other situations in which occasionally you'll have a baby born alive, and that's Edwards syndrome, or trisomy 18, and Pitot syndrome, trisomy 13. In both of these cases, babies born with this, and if they're alive, they'll typically live for a very short life, a couple of years. And people born with trisomy 18 or trisomy 13 have significant developmental delays, along with lots of other physical issues, heart issues, kidney issues, digestive tract abnormalities, delayed growth, etc. And most of these babies have what's called microcephaly, which is a small brain. So not too many autosomal aneuploidies out there in humans. But when we look at human aneuploidies of the sex chromosomes, it's much more common. And it's less damaging to the phenotype. Okay? So it turns out that about 1 out of every 1,000 females have an extra X chromosome. They have 3 X chromosomes, and they're healthy. They might not even know it. 1 out of every 2,500 girls is missing an X chromosome. Okay? She might not know it until maybe when she reaches reproductive age and has trouble conceiving a child. Some males have an extra X chromosome, so they have 47 total chromosomes and a sex complement of XXY. What's going on here? Oh, by the way, that number is something like 1 in every 500 males, and they're seemingly healthy. Well, think about this. Let's think about our female who has 3 X chromosomes. So this is maybe the nucleus of a cell with 3 X chromosomes. What happens to these X chromosomes? Well, in a female with 2 X chromosomes, what happens to one of the X chromosomes in every cell? It's inactivated and turns into a bar body. What do you think happens to a female with 3 X chromosomes? 2 of the X chromosomes are randomly inactivated in every cell, and she has 1 X chromosome that's active, just like an individual with 2 X chromosomes. Let's try the Klinefelter syndrome males. They have 2 X chromosomes and a Y chromosome, right? What happens here? Well, males don't normally have bar bodies. They don't normally have X inactivation, but guess what happens? In males with 2 X chromosomes, 1 of the X chromosomes in every cell is randomly inactivated and becomes a bar body. So a male that has 47 chromosomes and a sex chromosome complement of XXY will have 1 bar body in each of his cells. 1 of his X chromosomes becomes randomly inactivated. And so now you can start to see why people with an aneuploidy of a sex chromosome are probably pretty healthy. They have essentially the same amount of active X chromosome as everyone else. Polyploidy is when a situation in which you have more than 2 complete sets of chromosomes. So here we are, humans. Humans have 2 sets of chromosomes, right? We're diploid. Our gametes are haploid. Being polyploid means that you might be triploid. You might have 3 sets of chromosomes. Or tetraploid, having 4 sets of chromosomes. Apparently, plants can handle this. Lots of plants are triploid or tetraploid or octoploid. So our strawberries that we eat, I believe, are octoploid. Many of our apples that we eat are tetraploid or triploid. And bananas are triploid. This is kind of interesting. Bananas are interesting because have you ever seen a seed in a banana? You like open up a banana and you see a seed? Nah. Bananas don't make seeds. So they don't reproduce sexually? That's right. In order for a triploid to go through meiosis, think about that. Homologous chromosomes pair up in meiosis and then they separate nicely. Guess what? If you have 3 sets of chromosomes, how does meiosis happen? It's a disaster. And the gametes are a disaster. And bananas that are triploid can't make good gametes. They don't set seed. They don't reproduce sexually. So how do you get more bananas? You grow asexually. A big banana plantation will probably be genetically identical. Animals are much less likely to have polyploid situation. There are very few animals that are polyploid, but here's a cool exception. The blue-spotted salamander has a population that is all female. A whole population of all female blue-spotted salamanders. They are triploid. They produce triploid eggs that they lay that are unfertilized, but they develop into brand new baby female blue-spotted salamanders. It's an unusual strategy, but it seems to work in this particular subspecies of the blue-spotted salamander. Pretty cool. By the way, humans can, human embryos can be polyploid. That can be triploid. So occasionally, these babies do not develop normally and are miscarried. Okay, but if they are triploid, they would have 46 plus 23, 69 sets, 69 chromosomes in each cell. These fetuses do not develop very well and are always miscarried. Okay, so alterations in chromosome number, we're done with that. Alterations in chromosome structure. What if we change the structure of the chromosome? Will that affect the phenotype? Yes, right? Let's look at a couple examples of how. They're on the next slide. Okay, these are pictures that show what can happen what are some mechanisms by which we can alter the chromosome structure. So in the first example here, we've got a deletion, and the purple thing here represents a chromosome. All of the letters on the chromosome represent sections of the chromosome, pieces of the chromosome. And in the deletion, a piece of the chromosome is lost. This could happen accidentally anytime or during replication. There's a nice example of Cri-du-chat in the book that you might want to read about. I want to mention ring chromosome 13, and that's on the next slide, and we'll come back to this. So here's an example of a deletion. Ring chromosome 13 is a syndrome that my neighbor girl has. She is 18 or 19 and has grown up alongside my triplets, and she's a wonderful, beautiful young woman who is talented and independent and is working hard to become more and more independent. But she has ring chromosome 13. It's really rare. So she has 2 copies of chromosome 13, just like everybody else. One of the copies is linear and totally normal. The other copy turned out it looks like a little circle or a little ring. What happened? At some point during development, maybe in one of the gametes, maybe early in development, deletions happened at the ends of chromosome 13. Both ends broke off. And what that leads to are sticky ends. Usually they'll just come back together again. But in the case of ring chromosome 13, pieces of the chromosome break off and are lost. And what's left comes together in a circle. So one of her chromosomes is in a circle, and there's some genes missing from the ends of the chromosome. People with ring chromosome 13 have a huge set of phenotypic effects, but they vary somewhat depending on how many genes are lost as a result of this deletion. Duplication. So sometimes a piece of genetic material is accidentally duplicated. Oops, we got extras. We got extra pieces. So here's a good example of that, Fragile X syndrome. I want to tell you a little bit about that, and then we'll come back and look at these other two things. And there's a lot of information on this slide. We don't need to look at it all, but the basic idea is Fragile X syndrome is the most common inherited form of a cognitive delay. And it's the result of a duplication in one gene. There's a gene called the FMR1 gene on the X chromosome. You can see its location here on the long arm of chromosome X. That's called the Q arm, by the way. And this gene normally codes for a protein that's involved in the brain, and it produces, it's somehow involved in in regulating protein synthesis. Not exactly sure what it does, but it seems to be critical. Because in some individuals, okay, let's just start here. In this gene, there is a nucleotide triplet repeat. So there are 3 nucleotides, CGG, cytosine, guanine, guanine, that are repeated in the gene. Most people have a repeat of about 30 times. That triplet sequence is repeated 30 times. But some people have it repeated 100 times, or 200 times, or 800 times. And the individuals who have a lot of repeats, a lot of duplication in this gene, have problems. The gene doesn't work. It gets turned off, and then the protein isn't made, and this leads to a variety of symptoms, problems in the brain cells. That leads to learning disabilities, cognitive and mental disabilities, autistic-like behaviors, and a variety of other things. So this duplication is what leads to, in a single gene, is what leads to some of the symptoms. This slide here just shows you that the duplication is actually, or the repeat is actually unstable in some people. So most people have, 6 to 45 repeats. People with the full mutation have greater than 200. And then there's some people kind of in the middle, and they're usually not affected. Okay, males tend to be affected more quickly because they have only one X chromosome. Okay, that's an example of a duplication. All right, let's go to number 3, an inversion. An inversion is when a piece of the genetic material, a piece of a chromosome, is cut and then flipped around and then put back in place. And so you have all the parts, but they're in a little bit messed up order. It turns out that about about 2% of us humans have an inversion in one of our chromosomes, and we don't even know it. Usually it has very little effect, but we might have one. Sometimes it affects fertility, because a chromosome that has an inversion might have trouble going through meiosis, and that might lead to gametes that have unusual chromosome numbers. Okay, one more thing, a translocation. So in this situation, we've got to deal with 2 chromosomes here. You see 2 chromosomes, and one of them is long, one is short. And in a translocation, a balanced translocation at least, a piece of one chromosome ends up on another chromosome. Okay, there's kind of a swap of genetic material, a translocation. And these are not homologous chromosomes that we're dealing with. Let's look at an example. See where we are. Okay. This is kind of a fun one, and it's a little bit complicated, but we'll see what we can do. An example of a balanced translocation goes something like this. This is called a Robertsonian translocation because it involves a special type of chromosome. So chromosomes have centromeres, right? Their centromere can either be in the middle or it can be on the end. In an acrocentric chromosome, the centromere is so close to the end that the part of the chromosome that's above the centromere here there's not even any genes on it. Okay, a Robertsonian translocation is a translocation involving 2 acrocentric chromosomes. So let's think about this. Suppose we have chromosome 14 and chromosome 21. They're both acrocentric chromosomes. Sometimes these chromosomes become translocated. In other words, sometimes, like in the picture, this chromosome 14 becomes hooked to chromosome 21. At least the long arms of chromosome 14 and chromosome 21 become hooked together. The short arms become hooked together too, but that is just lost. Okay? So an individual might not even know it, but they might have a Robertsonian translocation of chromosome 14 and 21. So they have two copies of chromosome 14 and 2 copies of chromosome 21, just like everybody else. It's just that one of their copies is stuck together. Some people don't even notice it. Now, if you happen to have this particular balanced translocation, you are at higher risk of having a child with Down syndrome. A child with Down syndrome gets an extra copy of chromosome 21. This is an unusual situation of familial Down syndrome, or Down syndrome that is inherited. Most people who have a child with Down syndrome, in most cases, it's not a familial disorder. It's a result of non-disjunction that happens at random in an egg or a sperm. But this is kind of an unusual situation. So if a young couple has a child with Down syndrome, or maybe they have 2 children with Down syndrome, they might get tested to see if they have this balanced translocation, because it puts you at higher risk for having a child with Down syndrome. Let's take a look at how on the next slide. Okay, we've got Josh and Dawn here. Josh has two copies of chromosome 14, 2 copies of 21. He doesn't have a translocation. Okay, Dawn has 2 copies of chromosome 14, 2 copies of chromosome 21, but she has the translocation we were just talking about. She's healthy. She's perfectly fine. Now, they have a child named Tyler. What has Tyler inherited? Well, from Dad, Tyler inherits chromosome 14, 1 copy in, chromosome 21. Okay? His sperm have 1 copy of each. Dawn, her eggs have some trouble going through meiosis. Making the gametes might lead to some problems. How does this chromosome pair up during meiosis? Does it pair up with this guy? Does it pair up with this guy? What's going on? Well, that's why her egg could have an unusual number of chromosome 21s. In her egg, she could donate the translocated 14 plus 21 plus another chromosome 21. So in her egg, she might have an extra copy of chromosome 21. When her egg meets up with Josh's sperm, we would then have 2 copies of chromosome 14. That's fine. But 3 copies of chromosome 21. So this child, Tyler, has trisomy 21 or Down syndrome. This is unusual, but I just wanted to show you an example of a translocation. Okay. The rest of chapter 15 is fascinating. There's a section on imprinting behavior. Totally cool. I'd love for you to do that, but we're not going to test on it. I have X's here because you don't need to know this material, but you can check it out. Okay, a couple of YouTube videos to watch there. Another topic that we're not covering is the inheritance of organelle genes. So far, we've been talking about genes in the nucleus, but some organelles have their own genetic material. Which ones do? Well, in animals, mitochondria do. In plants, mitochondria and chloroplasts both have their own genetic material. That's called extranuclear, outside of the nucleus. And so how are these inherited? The patterns of inheritance of mitochondrial genes is different than nuclear genes. So read up on it if you're excited about it. Please do. This is a pedigree of a disease that's carried as That's a disease in which the gene in a gene in the mitochondria is affected. And this is just kind of a cool slide, blah, blah, blah. Okay, we're here at the end of chapter 15. The fun thing that you should watch is this kind of nerdy guy singing a biologist's Mother's Day video. It warms the cockles of my heart. So watch it and enjoy. And now you finished chapter 15. You should be able to do all of the problems on our problem set, our written assignment number 3. Should be able to complete all of those. I will help you with those. If you need help, you can email me or you can call me, or I possibly will have some virtual office hours that we can work through some of those problems. So at this point, you should be working hard to get those done. There we go. That's all we have. So let me get out of here and we will say goodbye. Okay? All right, I think that's it. We'll talk to you later. Bye.